Chromatography is a laboratory technique that is used to separate components of a mixture, molecules present in a mixture, using the property that is differential migration through a porous medium. There are two basic phases used in chromatography, that is stationary phase and a mobile phase. And it's basically, it is based on the affinity of these molecules to be separated either towards the stationary phase or towards the mobile phase, that these molecules get separated. Means the molecules which are having a greater affinity towards stationary phase will be held back or move slowly only, whereas the molecules which are having a greater affinity towards mobile phase will be carried by the mobile phase and will travel further. Now, it is basically it's the differential distribution of the molecules based on the affinity towards stationary or mobile phase. All the different types of chromatographies are having the same principle or working on the same principle, and the chromatographies are categorized into partition chromatography, adsorption chromatography, molecular exclusion chromatography, affinity chromatography, and ion exchange chromatography. And there are more mechanized advanced type of chromatographic techniques also, like HPLC and gas chromatography. Adsorption chromatography utilizes a solid stationary phase and a liquid mobile phase. The equilibration between mobile and stationary phase accounts for the separation of different solutes. The examples are thin layer chromatography and column chromatography. We can see how thin layer chromatography, TLC, is actually done in a lab. The materials required are a clean chromatography chamber in which the mobile phase solvent is to be taken. We are using amino acids as solutes in this experiment, and popularly used solvent mixture for separation of amino acids is butanol, acetic acid, distilled water mixture in the ratio 4:1:5. The stationary phase used is a thin layer of silica pasted on an aluminium paper. This is commercially available or can be prepared in the lab also. From silica gel, calcium sulfate in acetate buffer, which is spread on a glass plate, a clean glass plate, dried and activated at 110 degrees Celsius for around 1 hour in an oven. In any case, the TLC plate is ideally, it should be of 5 centimeters width and 15 centimeters length. The solvent mixture, butanol, acetic acid, water mixture, is added to the chromatography chamber, and this has to be kept closed for some time for vapor saturation inside it. And we have taken two types of chambers here. You can use whichever one which is convenient for use. Now, to start with, draw a pencil line about 2 centimeters from one of the edges and parallel to that edge. Never use a pen for this. And two equidistant spots are also marked on this line, 2 centimeters apart with the same pencil. The 1% amino acid solutions are used here. And one is a mixture of two non-amino acids, and the other is a test solution containing only one amino acid. And a small concentrated spot of solution that contains a sample is applied to the points using a capillary tube for maximum precision. Like the first is from the mixture, like this, and then the test sample is also added. The sample is absorbed onto the plate and may form interactions with it, and the paper is now allowed to dry. The dried paper is dipped into the chamber containing mobile phase solvent mixture, and care must be taken that the spots are above the surface of the solvent. And afterwards, the container is to be closed, just to be sealed. The solvent moves up the plate by capillary action, which occurs as a result of the attraction of the solvent molecules to the plate. And as the solvent rises through the plate, it meets and dissolves the sample mixture, which then will travel up the plate with the solvent at different rates due to differences in affinities. And approximately after 1 hour, when the solvent front reaches 2 centimeters away from the top, you can take the plate out like this, and you can keep it though, and mark the solvent front with a pencil. Again, I do not use a pen here, and allow the plate to dry in air or oven at 45 degrees Celsius. The solvent front is marked, and afterwards the solutes can be visualized using 1% ninhydrin for detection of amino acids, which is sprayed on the plate and the plate is dried in air or hot air oven. Now you can see two spots there representing two amino acids in the first point and a single spot in the test applied. The second point, the first spots are a bit more reddish purple in color. That depends on the type of amino acid, and central points are made to enable measurement of the distance moved as well as the distance from the point of application to solvent front is measured. Identification of amino acids are done by retardation factor or retention ratio given by the symbol RF, which is the ratio the distance moved by solute to the distance moved by solvent front. The RF value here for test spot is 4.5. This is divided by 12 centimeters. The distance moved by the solvent front gives an RF value of 0.375 for the test spot. You can calculate the other value yourselves on the data. The silica stationary phase is very polar and adsorb polar analytes more strongly, preventing them from traveling up the plate. Non-polar substances tend to move faster up the plate in TLC. Apart from ninhydrin, we can use some other agents also for visualization of the spots. Now, one is iodine vapor, which is sprayed onto the plate, and we can see the spots in a dark yellow to brownish color. Fluorescent compounds can also be used. One of the most popular compounds used is manganese activated zinc silicate, which is added into the adsorbent, and finally we can see the color in the black light, that is UV 254, and the background will appear light green, and the spots can be visualized as you can see. Now the applications of TLC. Now TLC is used to purify samples as well as to identify a number of biomolecules like acids, alcohols, amines, antibiotics, alkaloids. Now apart from amino acids that we used in our experiment. Now there are other molecules also like protein, ceramides, fatty acids can also be detected or identified, as well as pesticides and insecticides are detected in food and water. Another unique use is identification of phytochemicals in medicinal plants. So these are the applications or uses of TLC. Thank you for watching my video. Thanks very much.